Welcome back. Today we're going to be looking at the basics of the anatomy of the visual system. Now again, this can be new for people who are not familiar with this detailed anatomy, of course, and I encourage you again to supplement your textbook with this mini video, but what I'm going to do here is attempt to sort of make this as simple as possible while giving you the important details that are really necessary for understanding the anatomy and the details of how the visual system is going to be involved in producing our visual perceptions. So here in this figure, this is coming straight from the text. You'll see it in your book. You're seeing over here the eye, and then we're going to be zooming into the retina and looking at some cells that are really important for visual perception. Okay, but our first point of contact is a light wave, okay? In this case, we're looking at a key, okay, and light is going to pass through the cornea, okay, and that light wave is going to come into the eye. The pupil right here is surrounded by the iris, and the iris is going to limit the amount of light that's coming in. The lens is important for focusing the image, okay, and then basically visual data gets sent to a very important part of the eye known as the retina. Okay, and within the retina, we have our rods and our cones. These are special photoreceptors that help process light data and send that information to the rest of the brain. Also housed within the retina are a variety of other cells that are important for communication. Okay, so here I am zooming into the retina. Okay, I'm right here at the back of the eye. And if we zoom into the retina, we see a variety of cells. Here are our rods and cones. Okay, we see way more rods than cones in the retina. And then we have a variety of different cells, ganglion, amacrine, and bipolar cells. For now, all I want you to really know is that these cells facilitate communication between the photoreceptors and the rest of the visual system. Okay, so here we are again zooming in to this very organized setup of the human retina. And your really important cells are these photoreceptors that respond to different wavelengths of light, and that will be important for processing the visual data coming in and sending it to the rest of the brain. Okay, so I'm going to put that image up one more time, but you should be familiar now with the fact that the retina is critical for visual perception. It is the home of very important cells that help process visual data, and the main photoreceptors are those rods and those cones. So here we have an image just showing you the different wavelengths of light, and we're looking at the spectral sensitivity of the various cells. So short wavelengths of light, okay, tend to be perceived as blue, and as we get into more medium-sized wavelengths, the rods and cones that pick that information up are more in the green area, and longer wavelengths of light are perceived as red. This is an important figure to understand. The really important conceptual point is that your brain is taking a light wave, and with that light wave, it's interpreting it and producing a psychological perception of color. Okay, so color is a perception produced by your brain. We have a piece of energy coming in, a light wave, and your brain is producing the perception of color by interpreting those wavelengths. So just a little example there of the different wavelengths and how they're associated with perceptions of color. Now, here we have sort of the full circle picture here. I'm going to spend a minute or two walking through this figure, going into more depth beyond the retina of how visual perception is occurring. So here we have our left visual field. This is information coming in from the left side, and we have our right visual field. Okay, so our eyes are picking up that data. Okay, and here we are in the back of the retina, right, where the rods and the cones and some other cells are responsible for working with that visual information. Now, let's go ahead and zoom into this. Okay, this is sometimes where people get confused. We have the nasal hemiretina, okay, right here in blue. Okay, it's closer to the nose. Okay, this information is going to leave both eyes via the optic nerve. That's the nerve that sends visual information to the rest of the brain. Okay, we also have visual information coming out from the temporal side of the retina, okay, closer to the ears. Okay, now I'm not sure if you're aware that some of your visual information actually does a crisscross in your brain. The point of this crisscross is the optic chiasm. Now, information from the nasal hemiretina crosses over, okay, you can see that happening, and actually switches sides. So information from the left visual field, when we're looking at the nasal hemiretina, goes to the right hemisphere, and vice versa with the right eye, that information is going to go to the left hemisphere. That point of crisscross is the optic chiasm. Okay, the temporal hemiretina stays on its appropriate side, to just unpack the details there. Okay, so we have this really interesting crisscross of visual information. And why that's the case, why has our brain evolved to do that, is a really interesting question. There's different speculations out there about why the brain would sort of do this strange setup in terms of processing our visual information. But that's for another course that is going to focus more on the evolution of the brain and its structure. For our intents and purposes, after visual information reaches the optic chiasm, the nerve bundles are going to carry that information to the visual cortex. So here we are on the left and the right visual cortex. When we look in visual cortex, we'll see that there's an organization of layers that is going to further process visual data. Now, I want to hone in here on one detail. Okay, before this information comes all the way to visual cortex, it does get processed by the lateral geniculate nucleus. This is a portion of the thalamus that's very important for processing visual data. Okay, so after the optic chiasm, the visual information makes a pit stop, it gets processed by the LGN. Now here we are zooming into the LGN, and the LGN has different pathways. They're broken up into the parvocellular and the magnocellular pathways. That's some lingo you might have seen in the textbook. The key idea here is that parvocellular pathways, okay, process information from the cones. Remember, the cones are those photoreceptors that give you color, fine detail, and sharp vision. The magnocellular pathway is important for processing information from the rods, and those are responsible for dim vision, no color, sort of seeing in dim light. Okay, so the LGN is broken up into distinct layers that handles information from the rods and the cones. Okay, the layers are organized in that way. Once this processing occurs, information goes to primary visual cortex, where it will be further analyzed. Okay, so here's just another image. Okay, we have light coming in through the retina. It gets processed by the cells in the retina. Okay, gets sent out through the optic nerve to a part of the thalamus known as the lateral geniculate nucleus, which has a specific organization of layers that processes rod and cone data. And after that, a bundle of axons known as the optic radiation are going to send that visual data back to primary visual cortex, okay, the occipital lobe area. This is the general pathway for vision, and those are the general details that you should be familiar with. I'll include other videos below this expanding on this in different ways, and I highly suggest also reading over the portion of your textbook covering this process. That's all for now, and I'll see you in the next lecture.